This is Jose Merino, Editor-in-Chief of the Neurology family of journals. The Neurology podcast provides practical information to neurologists and other clinicians to help them provide better care for their patients. Thanks for listening and have a great week.
Hey, this is Jason Crowell from Norton Neuroscience Institute in Louisville, Kentucky. Thanks for listening to today's Neurology podcast. Today we have Subhojit Roy joining us. Subhojit is a professor of pathology and neurosciences at the University of California, San Diego. And today we're talking to him about a paper that he was a co-author on that was published in Neuron, December 20th, 2023. The title of the paper is Serine-129 Phosphorylation of α-Synuclein is an activity-dependent trigger for physiologic protein-protein interactions and synaptic function. Subhojit, thank you for joining us today.
It's great to be here. Thank you.
So we will do a little bit of background here before we talk about what you report in your paper. So I was just telling you a second ago before we started recording, I'm a movement disorder specialist. So I see lots of patients with Parkinson's disease and diseases that we describe as synucleinopathies. So I'm constantly answering questions from patients about α-Synuclein or using α-Synuclein when I'm trying to explain Parkinson's disease. But let's start there. What is α-Synuclein? What do we know about α-Synuclein and its role in neurons?
So if you think about one protein in Parkinson's, it's basically this protein α-Synuclein. So it's like the Michael Jordan of Parkinson's. I mean, there are many other proteins that are important, but there is not even a debate that this α-Synuclein is, the most important. And the reasons for this is essentially the fact that, as you know, all patients, Parkinson's disease, have accumulations of the α-Synuclein protein, their neurons, and also glia, depending on the disease. And then human genetics has given us a huge clue that mutations and even duplications and triplications of the α-Synuclein gene causes Parkinson's disease. And incredibly, the number of copies of α-Synuclein in this patient is dependent. So if you have, normally you would have 2 copies. If you have 3 copies, you have early onset Parkinson's. And if you have, more than that, you have more early onset. So there is just this incredible amount of evidence out there that this protein is important for Parkinson's disease. So normally this protein is quite abundant in the brain. So it's one of the most abundant synaptic proteins in the brain. So we know that it lives normally, mostly in the synapse. So given that there's been a huge amount of interest to find out what this protein normally does in the brain. So the idea is that if we really understand what the normal function is, then we would hold into how the disruption is happening in disease. And so that's where lab comes in. We've been working for over 10 years now on the normal function of α-Synuclein. We've slowly cut away the function. So in a nutshell, what we and my colleagues have found that α-Synuclein seems to be sort of like a brake or a clutch for neurotransmitter release. So as you know, the main function of neurons is to fire and communicate with other neurons. So it turns out that if you have slight excess of α-Synuclein in neurons, then you have a suppression of the neurotransmitter release, and vice versa. If you take it out, then there's more. So I feel at least there's a little bit of consensus in the field that the normal function of α-Synuclein is to regulate the extent of neurotransmitter release.
And just to clarify, we're talking about normal α-Synuclein, correct? So I think most of our listeners will be familiar with phosphorylated α-Synuclein, which is when this protein misfolds. Is that fair to say?
That's a great point. So just like you said, when the protein is phosphorylated at this particular site, which is the serine-129, we'll just call it phosphorylation. So when the protein is phosphorylated, then almost all the synuclein that's present in Parkinson's disease, that's aggregates in Parkinson's disease, is actually phosphorylated at the serine-129 site. So yes, the dogma in the field has always been that serine phosphorylation is just present in pathology, but it's actually a trigger of pathology. And so this has been prevalent. And what our data really shows is that this phosphorylation site actually has a normal role in the function of the protein.
I think it's fair to say that there's pretty good debate about whether or not this phosphorylated α-Synuclein is simply associated with Parkinson's disease or if it is causative, right? Is it contributory to the disease or is it something that's a byproduct of another process going on? Do you agree that that's a debate in the world of neuroscience right now? Is that fair to say?
There's been a debate for a long time that, yes, this phosphorylation is found in disease, but whether it's actually a cure of the disease or just associated with pathology, yes. But for the most part, people actually have sort of assumed that the presence of the phosphorylation is pathogenic, I would say. And the reason I say that is that there's a lot of animal studies, for example, where people would induce some sort of synuclein misbehavior, and then their readout would be the phosphorylation. Now, what you said is very nuanced because for the last about 10 years or so, people have been actually trying to figure out whether this modification is pathologic or not. And essentially, the data has been completely confusing. So most of the studies, they found nothing. And then some of the studies, it did induce pathology. And in the other studies, it was actually protective. So yes, there was a lot of confusion when the regular experiments were done, where you just kind of either put in the phosphorylated synuclein or don't put in the phosphorylated synuclein. But I think the problem was that everybody was sort of approaching it from the simplistic view that I'm going to either put it in or not, and not sort of going back and thinking about if this protein phosphorylation has a normal function, then what would it really do in the neuron? I mean, I'm not saying we actually stumbled on it by accident, to be honest. But that's kind of how the story evolved because we were always interested in what synuclein is doing in the synapse. And we just happened to look at phosphorylation. And it just so turned out that we found that when we killed phosphorylation at the site, essentially the protein was not able to do anything.
Yeah, so let's keep going there. So in this paper, again, the title has to do with phosphorylated alpha-synuclein being a trigger for physiologic protein-protein interactions and synapse function. So what did you report that the role of phosphorylated alpha-synuclein might be?
We have assays over the years where we can look at synuclein function, alpha-synuclein function. And one of the things that alpha-synuclein does is if you just throw in a very modest amount of alpha-synuclein in neurons inside the synapses, there is a suppression of neurotransmission. So that's been known for a long time. We've been working for it for a long time. So in that setting, when we tested phosphorylation, which is basically that this serine site, you mutate it to alanine, which cannot be phosphorylated. And when we did that, it was a real surprise for us that the protein, which normally suppresses neurotransmitter release, was not able to do that at all. So we kind of started from there and went back that why did this protein not work? So over the past 3 or 4 years, we've been looking at functional binding partners for alpha-synuclein. So the first thing we thought was whether the phosphorylation was actually affecting the binding of synuclein with its functional partners. And we were, to be honest, like completely stunned to see that this was essentially like a yes or no answer. So if phosphorylation happens at this site, then synuclein can bind to its functional partners. You can think about these functional partners as friends of synuclein that are absolutely necessary for synuclein to function. So when synuclein is phosphorylated at the site, it can bind to all its friends. But if it is not phosphorylated, it cannot bind. So nature has this mechanism in there to help synuclein do its normal job, which is suppress neurotransmission by binding to its functional partners, which is completely abolished when you prevent phosphorylation at this site. And that's really the crux of the paper.
So if phosphorylation is necessary for this protein to work like it's supposed to with its functional partners, as you say, then how could phosphorylation be pathologic?
That's a great question. So I don't have an answer yet, Jason, for this. But this is the conundrum that you kind of have a modification that is absolutely clearly present in pathology. There's no question about that. But you also have this modification in normal brains that is helping synuclein to do its normal job. So these, I mean, if I may say so, these are actually two facts. So somehow this normal function of synuclein, where it's just normally functionally, suppressing neurotransmitter release, kind of just binding to its functional partners, go happy and healthy. And something is happening along the way that is making synuclein hyperfunctional or just maybe some sort of a protective response. Maybe like one of the things, in neurodegeneration and also Parkinson's is hyperactivity of neurons. So if the neurons are sick, like if you're in the dopaminergic system, for example, we only look at the 5 surviving neurons that have alpha-synuclein, and we make a big song and dance that phosphorylated alpha-synuclein is important, right? But the fact is that a lot has happened to kill the 95% of the neurons before we even looked at it. So one possibility is that the phosphorylated synuclein is really a last-ditch, dying protective response of the neuron, where due to it's getting hyperactive, and synuclein is really trying very hard at this point to do its normal job. I think we don't have a clear answer to this yet, but I think the next step is to sort of slowly chip away at this bridge and see how the normal function of synuclein is related to what's happening in disease. So what's incredible is that there's some recent papers that have come out where people have carefully looked at what happens to the aggregation of phosphorylated synuclein. So synuclein, if you just take it and put it in a test tube and let it sit, it just aggregates over time, like many of these amyloidogenic proteins do. But turns out if you phosphorylated it, there's a massive suppression of this aggregation.
Now, wait a second. Wait a second. Now, I can't reconcile that either, Subhojit. Why? Because when we find it in these disease states, it is aggregated and it is phosphorylated.
So with the Lewy body, when you look at it with a regular light microscope, you just see the phosphosynuclein all over the place, right? But if you do a cryosection of the Lewy body or a cryo-EM of the Lewy body, the phosphorylated synuclein is actually like a shell around the Lewy body, and the center of the Lewy body is filled with different fibrils. I think we are going too much into the weeds. I'll have to have a whole different conversation with you on this sometime. But yeah, it is. I'm not denying the fact that the transition from a normal to abnormal is definitely something which is a black box at this point, But I feel good that at least we are not just, some of the old assumptions that we've had, for example, that, if you see something, it's definitely pathologic. Science is always like a deck of cards, right? I mean, there are 52 cards and you only have 5 of them right now, and they're not even in sequence. So it's like just you have to wait till the rest of the cards, come into play and things will become clearer slowly.
What are you all looking at now, trying to look at the next cards? What's next?
One of the curious things about phosphorylated synuclein is that if you look in the normal brain, you have this very high level of phosphorylation in only certain parts of the brain. And those are the superficial part of the brain. The olfactory cortex is like chock full of phosphosynuclein. And then very curiously, in the dopaminergic system, the system that's actually susceptible, which is the pars compacta, doesn't have much phosphorylated synuclein, but the pars reticulata does. So this is kind of a mystery as to this peculiar distribution of phosphorylated synuclein in the normal brain. That's where we are starting from, because we kind of want to know why is it present only in some of these locations. So one of the earliest sensory systems that is disrupted in PD is the olfactory system. So it's very curious to us as to why there would be so much of the synuclein in the olfactory system is phosphorylated. One of the hypotheses we have is that some of these systems are tonically active all the time. So maybe that's a protective system that evolution has built in to keep the neural activity at a basal level. So that's one of the things we are testing. We're also very interested to just do the very basic test first, whether the phosphorylation is neuroprotective or neurodegenerative, So for that, we are just expressing it in different systems where we can kind of look at the neurons and see how they respond to it.
Terrific. Well, this is a really interesting conversation. I think I have as many questions, if not more, than before we started. I guess that's the evidence of a good research question, and again, speaks to what we still have left to learn. I encourage our audience to read the paper. Again, this was published in Neuron on December the 20th, 2023. It was titled Serine-129 Phosphorylation of α-Synuclein is an activity-dependent trigger for physiologic protein-protein interactions and synaptic function. Subhojit, thank you for joining us. We really appreciate you taking the time to speak with us today.
Thank you, Jason.
This is Stacey Clardy, your podcast editor. If you've enjoyed the podcast, please take a few moments to subscribe, rate, and review the Neurology Podcast through Apple Podcasts, Google Podcasts, Spotify, or wherever you listen. And remember, you can always head to neurology.org/podcast for our full list of past episodes. Or you can also search by keyword in your podcast app for any neurology-specific topics you want to learn about.